and welcome everyone to this webinar on species interaction dynamics and ecological community structures in the context of gene drives. As this committee has been doing its work, we have drawn in experts from around the world to help us understand, as well as we can, this current state of the science as far as gene drives are concerned. And when we speak of the science, we speak broadly in terms of not just natural and physical sciences, but social sciences as well, and all the way on out into ethics and policy. Today, we're fortunate to have two distinguished individuals with us who are going to help us put in perspective the ecological issues associated with gene drives. So we're going to get started with Dr. David Lodge from the University of Notre Dame. David is a professor in biological sciences and director of the Environmental Change Initiative at the University of Notre Dame. His research focuses on ecological forecasting to better inform environmental risk assessments. He's really been a leader as far as understanding the biology of introduced organisms, and has worked really hard to help us understand exactly what the causes, effects, and how we might manage biological invasions. So with that, I'm going to turn it over to David. David, it's all yours.
Thank you very much, Jim. And thank you for the opportunity to be with you and the committee today, at least virtually. So the mandate that Professor Roderick and I were given was to think about species interactions and community dynamics. And as I've reflected on the fascinating and important and very challenging task of this committee, I've tried to distill what I know from my work that would be most relevant. And I've settled on focusing on what we've learned, the scientific community has learned from invasions and extinctions of species. And I will, during the next 25 minutes, try to draw out the most relevant lessons from what and I'm speaking as an ecologist who's worked on the management, policy, and economics interface, what we've learned over the last decades about invasions and extinctions of species. And I'm going to do this in 4 parts. The first 2 parts of my talk are going to address 2 different kinds of applications that I think the committee has considered already useful categorization as whether the gene drive might be used to modify a trait, or whether, in the second headline here, it might be used to eradicate a species. And then the latter part of my talk, I'll talk about what things we might be certain about and what things we might be uncertain about, and then explicitly about a few thoughts on risk assessment and risk management. But I'm going to focus now on applications that would modify a trait in a way that is not intended to lead toward eradication or extinction, but rather just change a locus in a species. And I want to suggest that there's a lot to be learned from the subdiscipline of invasion biology. And so I want to first make the case, which I don't want to overstate, but I want to make the case that when a gene drive might be used to modify a single or a few loci, that there's a similarity to alien invasive species. First of all, the gene drive application by definition makes the target species an invasive species in this sense. It's intended to be self-sustaining, and it's intended to spread. And those are the 2 key parts of what ecologists who study invasions see as the definition of an invasive species: self-sustaining and spreading. Now, normally, when we talk about invasive species, we're talking about species that also cause undesirable changes. But of course, in the applications of gene drive, at least the ones I'm going to consider, I'm considering only benevolent changes that are intended to be desirable. Now, the second part, alien, I want to make the case again, without overstating it, that when we speak of alien or non-indigenous species, we are thinking about species that are different from naturally occurring species. In the ordinary way in which we usually use these words, we're talking these species are different because they evolved in and came from somewhere else. They were transported to a new location where we refer to them as alien or non-indigenous. Now, obviously, in the gene drive applications, it's a little different. We're talking about making at least one change, but I wanted to remind the committee of Shingdar Sai's, what seemed to me a very important point from that earlier talk that you heard, that even when only one locus is the target, there are usually or perhaps always multiple loci. And so I just wanted to raise the possibility that there may be pleiotropic effects that would at least need to be considered. That is, interactions among changes at different loci that could lead to phenotypically or behaviorally important changes in the organism. But my main point to this point in my talk is to say that there's a huge discipline and a long history of studying alien invasive species, the ecology, the environmental effects, and the economic effects. I just show a few resources in that subdiscipline here. And I'll have time today only to draw out a few lessons, and I want to continue with thinking about what kinds of non-target effects might we expect, both on, if you will, the target species, that is the subject of the gene drive change, but also on closely related species. I want to continue with that pleiotropic idea just for a moment and suggest that, especially when we later on when I think about risk assessment, I'll invite you to think about how the changes that are made with a gene drive may not the impact of those may not be apparent in the laboratory only or in one kind of environment, but the express purpose of the applications, at least that I'm thinking about, would have the gene drive organism introduced into the environment. And what I'm suggesting with this first point here is that there may be effects that would not manifest themselves until the organism that's been modified is in interactions with other organisms in other kinds of environments. I'll say a little more about horizontal transmission because Nora Bazansky, my colleague at Notre Dame, covered that very well, and I have nothing to add there, but I wanted to remind you that it's a possibility if it's, even if it is of low probability. I do want to say a little bit more about hybridization, which Nora Bazansky and Allison Snow, I believe, also mentioned, and I want to explore that a little further. And I want to drive home the point with them that hybridization is very common among many kinds of usually closely related organisms. I've just got a couple of examples on this slide from work by myself and my own and my colleagues. I'll just mention a little bit about the top row, the first example on this slide, which has to do with the invasive rusty crayfish and how it readily hybridizes and with the result of viable hybrids and a lot of back crossing and changes in the native northern clearwater crayfish. In this case, these two species happen both to be native to North America, but have originally separate ranges. They've been brought into contact by human transport. So hybridization is very common in plants and in animals, and we cannot assume that it won't happen, particularly if the target organism would have any probability of coming into contact with closely related organisms. And we'll come back to that probability of sort of dispersal and contact as I go along. So now for the third blue headline on this slide, I want to now think about the potential for non-target effects more broadly, that is, on other species and on the environment. And I want to continue with this example of crayfish because it just happens to be one that I know well, but there are hundreds of such examples in the literature. And in this case, we have studied what has happened when the rusty crayfish has invaded lakes, hybridized with a native species, and ultimately replaced native species. And it's not just the effect on that closely related crayfish that happens. it's an effect on the entire ecosystem of the lake. I've got just a very simplified cartoon food web here on the left, and the vertical axis refers to what eats what, where the energy is coming from in terms of kind of organism. The horizontal axis refers to where in the lake the resource comes from. But the major point I want to show here is that the introduction merely of one organism, which differs very, very slightly from the organism that it replaces, that is one crayfish, the rusty crayfish replacing another extremely similar crayfish, results in very large ecological, ecosystem, and indeed economic changes in this example and others when this top predator becomes less abundant and it is the most economically valuable organism in this case study. Again, that's just one of many studies that I could pull out of that literature, a few books of which I showed you on an earlier slide. So effects from even small differences among organisms can have large effects. What's the predictability of those effects? With respect to the charge of this committee and thinking about recommendations for research or application in the future, I tried to think a little bit about what kinds of effects that I'm potentially negative, non-target, unintentional effects might be predictable. And unfortunately, from this, although it's a vast literature on the impacts of invasive species, so we know a lot about what those impacts can be, I would have to say that we do not yet have a theory or that would allow us to predict with any accuracy or precision what those effects would be ahead of time on the basis of the genetic differences between organisms. That remains an enormous challenge, which is made acute by the needs of risk assessment for gene drive organisms. Now, there is an area in which I think we have developed theory and ways of applying the theory that give us greater certainty and predictability, and that's the dispersal or the geographic spread of gene drive of organisms in general, and I would argue that it would apply as well to gene drive organisms as to any other organisms. So Alison Snow talked a little bit about what we might learn from a current population genetic structure. I want to emphasize a different point, a related point, which is that we know a lot about how organisms spread, both the natural dispersal mechanisms, and I will emphasize here, the anthropogenic dispersal mechanisms of organisms. We have an unfortunately long history of observing the accumulation of alien species. And again, I'm just offering you a few examples that I happen to know well here, that over time, in every ecosystem in which biologists have looked, we see more and more species. And my point in showing you this is to illustrate the incredible dispersal capabilities of organisms, both natural, but this in the examples I'm showing you reflect largely anthropogenic dispersal. I'll drive that home with this animation. If you look at the top, you'll see a time series, a year, and what you're looking at is the spread of two closely related species of mussel from Eurasia that arrived in the Great Lakes by ship, escaped through canals, started to spread across country on boats. We published a paper highlighting Lake Mead as a high probability site of initial invasion in the West. Indeed, that came to pass. That gave these mussels access to the extensive waterways of the West, and these mussels are still spreading through the West, and the political and natural resource leaders in the Columbia River Basin are taking steps to try to keep them out of there, because these mussels cause not only enormous ecological changes, but hundreds of millions of dollars annually in economic damages. That's just one, again, of many examples. I highlight a few more examples on this slide. My point is to say dispersal, global dispersal, is common for every kind of organism we know about, and every country for which we have records is subject to the same dispersal of organisms from elsewhere because of the globalization of transport and commerce, which I highlight in the next two slides. in this slide, you're looking at 24 hours of airline traffic. Now, when I look at an airplane, I think there goes a great flying arboretum and a great flying pet shop, because that's how many organisms disperse globally every day, either intentionally in the commerce in which living organisms are sold, like plants and living animals, pets and the like, but also unintentionally, including all the parasites and pathogens that traverse the globe every day on other organisms in planes or in ships. Here's 1 year's worth of global ship traffic. And if you watch 1 year worth of traffic, you will see that the Great Lakes or any other set of ports on the planet, or even in any individual port, is connected to every other port on the planet with no more than 5 ship voyages. So both the planes and the ships emphasize the rapid potential rate of global dispersal of any organism, organisms that exist now and organisms that might be altered through the application of gene drives. So I'm emphasizing that we have developed transportation models and other kinds of models that enable us, given a good deal of knowledge about an organism and how the vehicles by which it may move around, and including its natural dispersal, we have developed a good deal of predictability about where organisms might go. But the main message is they readily are transported everywhere on the planet under current conditions of policy and management. So far, I've talked about the kinds of considerations, lessons that we've learned from invasive alien species literature for those applications in which we've modified a trait with a gene drive. Now, many of those same lessons would apply, but I'm going to emphasize a few more that apply especially if the application is intended to eradicate a species. The target species might be modified with gene drive to eradicate it. So this might initially seem to be very uncontroversial if we're thinking about species that are obviously harmful, rats, mosquitoes that carry malaria. But even those cases in which the potential benefits are obvious and enormous, one of my points is to suggest that there nevertheless are non-target impacts that would not be trivial. Every species has, especially if it's an alien species, has populations of the same species somewhere else in its native range. So if I'm thinking here of applications that might have their goal eradicating an invasive species, rats on islands, Anopheles gambiae mosquitoes carrying malaria. But those species also have populations somewhere else, populations within reach of dispersal under normal circumstances, as I've already suggested with the examples of planes and ships. They also, either close by in the target population or more distantly somewhere else on the planet, have species with which they're closely related and therefore could hybridize and spread the gene drive beyond the target population, potentially beyond the target species, and beyond the target geography. Again, I'll use the example of crayfish just to keep a continuous thread of an example going here. There are 300 species of crayfish in North America. I emphasized earlier one crayfish, the rusty crayfish, which is harmful in regions of North America where it has invaded, but of course is native elsewhere. And that applies to many other species globally as well. So we have to think about non-target effects on populations of the same species and also on closely related species, not just in the same continent, but globally as well. Now, if the target is an invasive species, or any species for that matter, there's also something that, as I've reflected on this issue over the last few weeks, I think is important to consider, a topic on which I'm not an expert, but I believe could be quite important. And that is that if for gene drive applications that are the purpose of which is to drive a population extinct, there will be strong selection for parthenogenesis. And it is surprising, I think, to many, even biologists like me, how the wide range of taxa that exhibit parthenogenesis, that is reproduction with only one sex, usually female, I believe. And while in some of these, and I just illustrated a few of the kinds of taxonomic groups in which parthenogenesis is known to occur, in some of these it's very common, in others it's very rare. But under a gene drive to eradicate the sexually reproducing parts of a population, which is how the gene drive, in my understanding, would work, there would be a strong selection for parthenogenetic individuals, something to consider, I believe, as the committee goes forward. Now, I also want to think about these applications for extinctions and think a little bit more about non-target effects. We have an unusually, an unfortunately rich experience, we humans, in driving many species locally or even globally extinct. We've usually done that unintentionally. And the only point I want to make is that, again, there is a very, very rich literature. I'll highlight one example here in which the almost extinction of sea otters brought enormous changes to many other organisms in ocean communities in the Pacific Northwest of North America and Canada, and leading in recent years to a change in the diet of killer whales. These kinds of examples are rich in the literature, and they are also, unfortunately, rich in results that I don't think we could have predicted ahead of time, even with years of study of a food web and an ecosystem. I want to offer one final point under this heading, and that is that offer for the committee's consideration that, as I've reflected, I think the rate of population suppression by a gene drive designed to drive a population down or even extinct would be rapid relative to the rate by which other species in that ecosystem could acclimate or evolve to the absence of that species. And I say this because although we humans have driven many species extinct in the past, we have usually done so relatively slowly compared to what I think would occur in the kinds of applications of gene drive that I at least have heard about. So I think it would take some more thinking about what the implications of those rates would be. But I think the general point I would make here is that what we've learned from how ecosystems adjust to the removal of species in the past might not apply as fully as we might hope, because we might be removing a species much more rapidly than we have in the past if we use a gene drive. So far, I've talked about applications in which we might modify a trait, contrasted that with applications in which the goal would be eradication. And now I want to shift and think as I begin to wrap up about how we can be, what aspects of these things we might be certain about, what uncertain, and explicitly think a little bit more about risk assessment and risk management. So I think the one thing that I feel very confident about, based on the literature of the ecological literature of the additions and subtractions of species, is that there will certainly be unintended, unanticipated ecological, environmental, and economic impacts. What I think is unfortunately much less certain, for which there's high uncertainty, is predicting what the magnitude of those effects would be, or even their desirability of those unintended and unanticipated results. Now, shifting more explicitly to thinking about risk assessment and risk management, I want to emphasize that what I illustrated earlier, that dispersal, both natural and by anthropogenic means, is going to be common. I also want now to suggest that it's worth thinking in terms of risk assessment explicitly about whether modification or eradication might bring fewer or lesser risks in different circumstances. I'll illustrate that by looking at this example where if we think about mosquitoes, which are a commonly discussed target of eradication or at least the modification of vector competence, as I think about the potential non-target ecological effects, I believe what we might expect is that a goal to modify the vector competence of the mosquito might bring fewer non-target effects than a goal to eradicate the mosquitoes. And this is because mosquitoes are not only connected to humans, but they are also a food source and engaged in a food web that involves many other organisms, many of which have obvious or benefits to humans, and many of which we probably have no idea what benefits, if any, we humans derive. But my general point is it's worth thinking about for any goal how to minimize the non-target effects, and that involves perhaps at least a deliberate choice about modification or eradication of the species. So to wrap up, I've emphasized the dispersal mechanisms and how important they are to consider. I think that likelihood of dispersal both across both national or within nation political jurisdictions and across national boundaries suggests that the committee should think about policies, both national and international, that already address invasive species and think about those in the context of how applicable they might be to gene drive organisms. I've suggested that at least in some applications, trait modification might be less risky in terms of ecological non-target effects than eradication. Now, for risk assessment, I think that any case is going to require a rigorous assessment. And Allison Snow made this case, I think, well as well also. I think a risk assessment based on the ecological literature, that extensive knowledge of many examples in the past, unfortunately says that we still would require for each case, in order to gain any kind of confidence, a multi-generational, that is a long-term, multi-scale, and well-replicated trials in as close to a natural environment as possible, but of course an environment that would have to be fully contained. Islands have often been mentioned as possibilities, but again, think about those dispersal mechanisms and how readily organisms on islands, and think about how readily rats have gotten onto many islands around the planet and how they leave places and go to others, on ships, for example. So any risk assessment trial would have to consider containment of that location for the maximum lifespan, not just of active organisms, but also of resting stages. And so to conclude, in thinking about risk management, unfortunately, I've highlighted a number of risks that the ecological literature says are likely to occur. What about managing those? It strikes me that the proposals for fighting a gene drive release gone awry with another gene drive release are definitely worth pursuing and thinking about, but they seem a little optimistic. We have a lot of examples from the biocontrol literature that would suggest that fighting fire with fire can sometimes burn the house down. So that requires a cautious approach. In any field trial, whether it's a risk assessment or application phase, there's going to need to be surveillance. But the rich experience with invasive species says that this is very difficult. We could talk in Q&A about some ways in which this is done, but I don't think there's a silver bullet there, and there's definitely not anything cheap. The same for monitoring of non-target effects. What indicators would one choose? There's a rich ecological literature on indicators, which again suggests it's difficult to find a few cheaply measured things that tell you what you want to know. And we need a toolbox that's got gene drive reversal in it perhaps, but some other tools in it. And finally, thinking about as these things are done intentionally, where's the legal liability going to lie? Thank you. I know I need to quit, and I appreciate your attention And I'll stop there. Thank you.
Terrific. David, Audrey, can you hear me?
Yes, we can.
Okay, good. Thanks. Excellent. Thanks very much, David. That was a wonderful presentation with lots of really good points, lots of things for the committee to think about.